Okay. All right. So again, good morning. If I just let you in, good morning, good morning, good morning. Um, if you have not, and I know only three people have done so, if you have not turned in your Pick Your Poison project, please use the next 45 minutes to do so. Halfway through class, I will show a video. We're going to start the next unit on serology. So if you're finished, you want to look at the vocabulary page, the introductory page, and get familiar with the vocabulary. We are going to start serology today. So please use the first 45 minutes of class. If you used working on in class last time, you should be finished. Like I said, I've got three people who are already finished. Everybody could be finished. You only had to pick two poisons. All right. So again, next 45 minutes, and then I'm going to uh, introduce serology. If you finish, you submit your project, go to Canvas and look at the introductory page on serology. Get familiar with what is serology. I'll do a little biology in that we're going to look at what is blood and blood cells and what their function is and what we're going to be looking for as forensic pathologists. All right, being as we have a lot of people who need to come in, I'll say this all over again in the next 5 minutes. All right, so again, if you've already turned in your project, go to Canvas and introduce yourself to serology. Melissa, I see you on twice. Are you on two different devices? All right, again, good morning. Good morning. Some of you just joined. We are in the next 40 minutes now. We're going to complete the pick your poison activity, get that submitted, and then we're going to move on to serology. So if you are finished or when you finish, look at the vocabulary for serology, get to know what serology is. And we're going to have a little introduction to blood and the function and what we're looking for when we're forensic scientists. So about 9:15, we'll start talking about that. Remember, you only had to pick two poisons. You need to talk about how they're classified and then answer the questions that go from the outline for each poison. You want a picture that represents the poison. And so that's two pictures, one for each poison. You want to make sure that you have your references cited for each poison. Make it interesting looking. So follow the rubric. All right, so again, good morning. Good morning. If I just let you in, we are trying to get your project finished by 9:15, and then we're going to go on to serology. If you are already done, please look at the introductory page for serology in Canvas. Today's the last day you'll have class time to work on that. So please make sure you use it wisely and get it done. Emmeline, yes. Yes. And explain why. So again, folks, you have a topic. When you pull up that topic, so be household, food, animal, um, plant. When you pull that list up, you'll have a list of things that create poisons. Pick one from that list. It can, and then go through the checkpoints to make sure that you answer all of the points for that poison. With foods, the bacteria create a poison. And we got 30 more minutes to work on that, please. Okay. I just wanted to go over the rubric with you again. So before you submit your project so that you can get full points, make sure that you have checked with the rubric to make sure you've done everything that you need to and you didn't just give away your grade. All right, so you have picked your topic, either household or environmental, plant, animal, or food. You picked two of those. And then when you looked that up, you found a poison underneath one of those headings or two of those headings. And you, you are answered number one, it says sufficient and accurate information on 5 points. The 5 points are describe the poison, the type of poison, which includes the target organ. So what part of the body does it affect? Um, the, 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 how much of the sub, when it says level of toxicity, what, how much of it is poisonous? Is there a small amount that is trace? Is there a certain level that will kill you? Or there's a certain level that just might make you sick? The symptoms that you would cause the forensic anthropologist, not pathologist, to notice the symptoms so that they can decide that this is that poison. And 5, any additional information that you give. You might have a case that that was solved by identifying this poison. You can use as your 5th point, possible cures and treatments, lasting effects. Any of these things will add up to 5 points, or you can touch on all of them. But at least 5 of these things need to be included in your answer. 3 points. So if it's 15 points, that's 3 points a sentence, an illustration, and one that should um, illustrate the poison itself. What does it look like? Um, or what does a person who is suffering from this look like? It should be neat and easy to read, so you get 5 points for that, for being presented nicely. The same thing for the second poison. Okay, and again, you're getting presentation skills, 5 points, and your 2 references. Okay, so this is a 60-point project. So please, like I said, do not give away your grade. You should have at least 2 references, 1 for each poison, and they should be stated at the end of your video. Okay. All right, another 20 minutes, and then we're moving on to serology. Okay, this on there. All right, so when I do the grade, I'm going to have the rubric up, and on the point distribution will be in the notes section of the grade box. All right, I'm still here. I just need to be off camera for a few minutes. Can you make up the test? Um, Let me go in and open it up. I think it's closed. Um, why does it do me like that? Give me 1 minute, Kenzie. I have to change the date and give you a code. Okay, so today is the 20th. Actually, let me see if anybody else needs to do that. Shayla, are you here today? Nope. Okay. Again, please be using this time to get this project done so that we are not falling behind. We're almost done with this quarter. We don't want to have to be trying to turn in 5 assignments next week. It's just a minimum of two. If you have more references, that's fine. I might add too, folks, that a lot of times the picture that you select also needs a reference. So if you go to, if you click on, when you click on a picture on Google's images, if you click on the one that says view source, so it'll tell you where the picture came from, you can use the web address for that website for your picture. So yes, picture should also be cited as well. Unless you got the picture from that website that you got your information from, then you don't need a separate um, source. Hmm. Okay. So, Hmm. Okay, I figured as much because I know you were in. All right, a few more minutes, folks, and then I'm going to show the video on serology. Again, if you're already finished, please start looking at the vocabulary for serology in Canvas. That is our next topic. Okay, folks. I have checked the ones that have been submitted, which are not very many. And I like the fact that when I said I didn't have to tell you which poisons to take, that you all have very nicely so far selected a variety of poisons. So I'm happy about that. All right. So again, our next stop, so stop what you're doing, kind of give an ear. I did want to introduce serology. I do have a list of videos in the playlist that we're going to watch. I'm going to do two of them this morning. One basically is to give you a biological background on what blood is. And then we're going to look at it from a forensics point of view. All right, so give me 1 second to get this up.
On blood. This is notes number 1 of the serology unit. We are going to focus on blood, even though oftentimes during serology that does include other bodily fluids, but during our investigations we'll be focused primarily on blood. So let's talk about blood. First of all, the average human adult has approximately 10 pints of blood. So that would be about 5.5 liters. So if you can think of a 1 liter bottle or even a 2 liter bottle, kind of figure out what 5.5 liters would be. That's how much total blood the average adult has. Blood is actually made of 2 different parts. There are the cells, and then there's the liquid, which we call plasma. So we're going to talk about the 2 different parts. In order to understand how we use blood in forensics, we have to understand what it's made of and what those parts are, so you can understand how we then use them to potentially solve crimes. Blood cells are produced in the bone marrow, in the pelvis, and sometimes in the upper part of the femur. Your other bones do have a middle substance of bone marrow, but it's not active. It is active when you are developing in your mother's womb, and as you get closer and closer to birth, more and more of the small bones, the bone marrow shuts down, so that by the time you're an adult, the only bone marrow that's active is your pelvis, and again, sometimes the upper part of the femur, but primarily the pelvis. Just quick aside, so here in the pelvis, if you wanted to give someone a bone marrow transplant, because there are some diseases like leukemia, which is a cancer of your white blood cells, to cure that disease, they have to kill all the bone marrow because it's the part that's making the cancerous cells, and then transplant it with bone marrow from a healthy person. So they actually take a big needle, inject it, drill into the pelvis and extract some of the bone marrow, and then put it into someone else. Now there have been 1 or 2 cases where someone who has gotten a bone marrow transplant has gone on to commit crimes, and their DNA from their blood doesn't match the DNA from the rest of their body, because the bone marrow from the donor produces the blood cells in their blood, which is where we get the DNA, but their cheek cells and the other cells from the rest of their body have their own DNA. So there have been 1 or 2 cases like that in the United States where a criminal got away with it for a little bit until they figured out what was going on. Not a common thing at all, but it has happened. All right, in terms of the cells that are made in the bone marrow, The most common is called the erythrocyte. We call it the red blood cell. This is a super important cell because it carries oxygen. They look like little red donuts, except for there's no hole. In that middle right there is where the oxygen binds. There's a molecule called the hemoglobin, which your body makes, and it allows these red blood cells to pick up oxygen in the lungs and transport them throughout your body. Red blood cells do not have a nucleus. They lose their nucleus before they leave the bone marrow. And that's in order for them to be as flexible as possible to fit through all of your blood vessels without getting stuck. The nucleus would be a hard thing in the middle that might get them stuck. And if you get stuck in the middle of a, let's say, a capillary or a blood vessel, that would cause potentially a heart attack or a stroke, which would be something we don't want. So the red blood cells do not have a nucleus, and we cannot get DNA from red blood cells because they have no DNA in them. Other than mitochondrial DNA, they still do have mitochondria. We still can't get mitochondrial DNA. More than 99% of all the blood cells in your blood are red blood cells. They're that important. There is a test that you can look at to see how many red blood cells there are. It's called a hematocrit test. It basically takes the percentage of your total blood volume that's red blood cells. They do this test not usually to help solve crimes. They usually do it to see how healthy you are. But I think it is interesting that males on average have 45% of the total blood volume is red blood cells. So 99% of the cells are red blood cells. But when you take into account all the liquid portion, again, the plasma, it's about 45%. You can see the other cells are this little white line there. Females are a little lower, and the reason females have a lower count of red blood cells is because women menstruate. So because of a monthly period, they're losing blood, and females, your bodies have to work that much harder to create new red blood cells on a constant basis, where males do not. I guess they could use that test to differentiate between male versus female, but it's unlikely. It's much more of a clinical test for disease. Anyway, the next kind of blood cell is the leukocyte or white blood cell. And leukocytes, you can see they're a little larger than red blood cell. They're also generally more circular, and they protect against infection. There's several different types. They have several different roles in the immune system, but they are key to the immune system, one of the biggest keys. And they do have a nucleus, and when we test blood for DNA, it is in the white blood cells that we are getting that DNA from.
Commercial time.
The third cell is actually not a full cell. It's called platelets, and they are cell fragments, and they are involved in clotting. So you can see there are a whole bunch of little platelets, and essentially when you get cut, there are chemicals in your cells that get released that activate the platelets to make them get sticky. And they get sticky and they stick together, and they form a barrier that blocks the red blood cells. And once you get enough red blood cells stacked up against this sticky platelet mesh, then that stops the bleeding. So that is how clotting works. Platelets are the main thing involved. Okay, so that covers the three cells: red blood cells, white blood cells, and platelets. Now let's look at the plasma. The plasma is the liquid portion of your blood, and there are a large number of things in your blood, of organic and inorganic substances that are dissolved in essentially water. There are three different main proteins that are in your blood, and so I'm going to tell you what those three are, and then we're done. The first two we're going to talk about together. One's called the albumins, the other is the globulins. They essentially work the same way, but they're structurally very different, which is why they have two different names. There's a picture of an albumin, and here's a globulin. You can see they look nothing alike, but they do the same kind of jobs in the blood. So some of the functions that they do, they can act as buffers. And if you remember from chemistry, a buffer is a substance that keeps the pH of a liquid from going too high or too low. So it keeps the pH in a steady range. They also often bind and transport other things in the plasma to move them around the body. Things like lipids and hormones and vitamins and metals. Like if you have iron in your blood, most of that iron is in red blood cells, but you could have some iron, but it's not just going to be like an iron molecule floating around the blood. It's going to get attached to an albumin or a globulin that then transports it around your blood. Same thing with hormones. Some albumins and globulins have a little bit of a factor in that clotting response with platelets, but not much. Some albumins and globulins act as enzymes, and if you remember from biology, an enzyme is a molecule which helps a chemical reaction happen. Some of the albumins and globulins are hormones themselves, and some of them are antibodies, and antibodies are one of the key structures in your body responsible for your immune system. Along with the white blood cells, antibodies are key. We'll get more into antibodies in another notes. The final type of protein in your plasma is called fibrinogen. This is the other key component in clotting. They act like little fibers. So when the platelets get activated and get sticky, the fibrinogen also gets activated and it gets stringy. And between the stringing and the sticky, it's able to make that nice web mesh to allow you to have clotting so that you can form scabs and things like that. So that's it. We've got your basic idea of what blood is made of, the plasma and the cells. And starting next notes, we'll start talking about what kind of tests we can do in order to use that knowledge of what blood is into solving crimes. Thank you for listening. I'll talk to you next time.
Okay, so we will look at that next part next time, but I did want to switch to this other one. We have just enough time left to watch this one.
Check this out. I'm going to show you how I afforded this car. Okay, you ready? All right. $118 for car insurance. That's how much I was paying. Okay, I picked.
All right, forensic students, we are starting on our blood evidence section of forensic evidence, which is Unit 2. And we're going to cover blood in a couple different lessons. So today, specifically, we're going to focus on forensic serology and then a division of forensic serology, which is going to be blood typing. We're going to talk a little bit about blood typing and how it can be applied to forensics. And then in future lessons, we're going to move more into the physics of blood and how it moves and how it travels. And that's going to be in our blood spatter lessons. So before we jump into blood typing, you need to know that there is a branch of forensic investigation that is the heart of crime solving, even more so than fingerprints or hair analysis or fiber analysis, and that is forensic serology. So forensic serology, when combined with the incredible science of DNA analysis, it can provide the indisputable piece of evidence that places a suspect at the scene of a crime and ultimately puts them behind bars. It is a hardcore science, and it is the hardcore science behind thousands of real-life cold cases that have been solved. And so it is extremely important to talk about. We do not need to overlook forensic serology. And if you're looking for a definition, I've got one on the screen. Might be a good idea to write this down. So serology in general is just a term that's used to describe really a broad range of laboratory tests that uses chemical reactions between blood serum and other bodily fluids and then reagents. So we're going to talk about some of those chemical reactions today. Now, a serology lab is going to cover a lot of different areas, and I've listed some of those areas on the screen. So you can have a forensic serology center or forensic serology lab, and within that laboratory, there's going to be different divisions. So you can have an area that specializes in blood typing, an area of that laboratory that characterizes unknown blood, an area that looks specifically at spatter patterns or blood stain patterns, and they do an analysis or crime scene reconstruction. You can have a division for paternity testing and then semen identification in rape cases. And then, of course, you can have just strictly DNA identification. All right, so there are 3 different questions that has to be answered by a forensic investigator when they're working a crime scene and they have found something that they believe could be blood. So the first question a crime scene investigator is going to ask is this actually blood? And they're not going to have time to swab that sample and send it off and wait days or weeks or months for those test results to come back. So an investigator is going to have a ready to go on the scene test that they can use to test to determine if something is indeed blood or not. They call these tests presumptive blood tests, and you can see on the screen there's different versions of these tests. But again, a presumptive blood test is an on-the-scene qualitative analysis that just identifies or confirms the presence or absence of a substance in a sample. So usually these presumptive blood tests use a chemical reaction to identify a positive result. So you can see one of the presumptive blood tests that are often used by investigators is something known as the Kastle-Meyer test. And this is a chemical, or Kastle-Meyer reagent is a chemical indicator, specifically phenolphthalein, that reacts with hemoglobin in blood. If it detects that hemoglobin, it will indicate the presence of blood by turning a bright, like magenta color, or like a really bright pink color. And I've got a picture of that in like the next couple of slides. I'll show you that. You can also have a presumptive blood test like luminol. This is one that if you've watched shows like CSI or Criminal Minds, you've probably seen the investigators use something like luminol. But again, luminol works the same way. It is a reagent that's just added to what investigators believe to be blood, and through chemical analysis, it will indicate the presence of blood or not. Specifically, luminol can be added to latent blood stains. Those are stains that can't be seen with the naked eye. And the luminol will actually bioluminesce or glow if it detects blood. So it is great for blood stains that have been cleaned. So if somebody's cleaned a crime scene to try to make blood disappear, luminol can detect that blood. Now, the second question that's going to be asked by investigators, after they confirm that they have blood, they're going to want to confirm that they have actual human blood, and then they're going to want to sample that blood, send it off for DNA analysis, and then they might also use it for blood typing purposes, which we'll also talk about today. All right, so this is just an example of the luminol test, which again is a presumptive blood test. Can be done on the scene. You don't have to wait days or weeks for the results to come back. And you just need to know that blood contains, or red blood cells contain, hemoglobin and iron, and the iron is actually what reacts with the luminol. And with a UV light, it can be made to glow. And so you can see that in the picture here.
All right, I'm going to pause it here and we'll pick up on this next class. So we got about 3 more minutes left to class. So please make sure that you are indeed making sure that your PowerPoint is submitted. And again, please make sure that you look at the rubric so that you get all the points you deserve. And I looked at one just now that did not, while it was very well done, very detailed, you had to actually read it to figure out, is this what the heading was, whether it was household, plant, animal, or food. So please make sure that you classify what your poison is, and then answer all of these, or at least five of these points. Some of you have done very nicely and included a case study, and I like that. That was good. So thank you, thank you. Those that I've read already are well done. All right, so again, today's the last day for this one, so please make sure you have it turned in today. If you are already completed, you're good to go for today. Stay warm. It is freezing out there and it's not going to get any better. So, at least it is up here in the North Georgia. I mean, you're kind of South Georgia, but it's still cold. So have a good weekend. And please, please, please get your projects turned in.